Hello, this is Mike Klepser, and today we're going to be talking about oral therapies for SARS-CoV-2. By the end of the presentation, I'd like you to be able to summarize the life cycle of SARS-CoV-2 and identify possible target sites for drug development. Secondly, we'd like to summarize the clinical and safety data for Molnupiravir and Nirmatrelvir ritonavir, or Paxlovid. First, regarding coronavirus replication, SARS-CoV-2 is an enveloped, positive-sense, single-stranded RNA. The primary protein that's responsible for host cell attachment internalization is the S protein, which is expressed on the virus surface. The S proteins bind to receptors, and then cleavage of the S protein permits fusion with the cell. Following entry into the host cell and uncoating, 2 polyproteins are produced and then cleaved into 16 non-structural proteins by viral proteases. The primary protease, Mpro, releases the majority of non-structural proteins from the polyproteins and is essential for the viral life cycle. This makes the Mpro enzyme an attractive drug target. Viral genomic replication is facilitated by an RNA-dependent RNA polymerase that resides in a non-structural protein, number 12. Antivirals often target viral polymerases and function as nucleoside analogs that terminate RNA chain elongation. However, such chain-terminating antivirals are generally not effective against SARS-CoV-2 because coronaviruses carry an exonucleic proofreading activity that can remove misincorporated nucleotides from the nascent RNA 3'N. The nucleoside analog remdesivir can circumvent proofreading because its incorporation does not terminate elongation, but only stalls RNA-dependent RNA polymerase after the addition of 3 more nucleotides. The subsequent structural proteins that are produced are assembled and form the new virions, which then are released from the host cell. With respect to the current therapies that we want to talk about for SARS-CoV-2, we'll be talking about 1 protease inhibitor and 1 RNA-dependent RNA polymerase inhibitor. These 2 medications are Nirmatrelvir and Molnupiravir. Molnupiravir is a nucleoside analog that inhibits viral replication by promoting widespread mutations in the viral replication by inhibiting RNA-dependent RNA polymerase. Molnupiravir acts as a mutagenizing agent that causes an error catastrophe during viral replication. It's hypothesized that this occurs in 2 steps. First, when Molnupiravir prodrug enters the cell, it's converted to NHC triphosphate, which can be used by the RNA-dependent RNA polymerase of SARS-CoV-2 as a substrate instead of cytosine triphosphate or uracil triphosphate. Therefore, in the first step, the RNA-dependent RNA polymerase is predicted to frequently incorporate the drug or the NHC monophosphate instead of cytosine or uracil when it uses the positive stranded genomic RNA as a template to synthesize negative strand genomic RNA. In a second step, the resulting Molnupiravir containing RNA can then be used as a template for the synthesis of positive stranded genomic RNA or positive stranded subgenomic mRNA. And the presence of the Molnupiravir NHC in this negative stranded genomic RNA then leads to mutations in the positive stranded RNA products, which do not support the formation of intact new viruses. And as predicted through a number of studies, this then produces the error catastrophe, which is hypothesized to be the mechanism of action of Molnupiravir. Regarding the pharmacokinetics of Molnupiravir, following administration of 800 milligrams every 12 hours, the area under the curve from 0 to 12 hours is roughly 8,260 nanograms hours per milliliter. The Cmax is about 3,000 nanograms per milliliter, and the Cmin is about 31. Administration of a single dose of Molnupiravir with a high-fat meal does result in about a 35% reduction in the production of NHC. However, the Cmax and the AUC are not significantly affected. Regarding the elimination, renal and hepatic modalities are not major routes of elimination for Molnupiravir or its metabolites, and therefore the drug-drug interaction potential with this medication and its metabolites is low. The half-life of NHC, or the active component of Molnupiravir, is approximately 3.3 hours. And the fraction of the dose that's excreted in the urine was less than 3% in healthy individuals. Again, highlighting the fact that renal elimination is not the major route of elimination for Molnupiravir or NHC. Because of the mechanism of action, there has been concerns regarding teratogenicity and bone marrow suppression. In a number of animal studies, there did appear to be conflicting data. Some studies in dogs did demonstrate reversible bone marrow toxicity affecting hematopoietic cell lines when the animals were exposed to drug concentrations that were roughly about 0.4 times the human exposure. There were mild decreases that resulted in blood cells and platelet counts after 7 days of Molnupiravir, and there were more severe hematological changes after 14 days of treatment in the canine models. Neither bone marrow or hematological toxicity was noted in murine models. There does appear to be some bone and cartilage toxicity when looking at physical and epithelial growth cartilage. These were generally observed with long durations of therapy at very high levels of exposure. These same sorts of toxicities and changes were not observed in shorter studies. Regarding fetal development in rats and rabbits, there were developmental toxicities that did occur when animals were exposed to high levels of NHC. In rabbits, there was also developmental toxicity resulting in reduced fetal weight, but again, these were at very high levels of exposure. With respect to preclinical data, studies reveal that Molnupiravir possesses broad-spectrum antiviral activity against coronavirus, including SARS-CoV-2. Importantly, it is noted that there is a high barrier to resistance against this medication. However, owing to the mechanism of action of Molnupiravir, there is a theoretical concern that Molnupiravir could generate mutations in the coronavirus genome that could lead to the emergence of new variants of concern. And as we'll show shortly in the clinical data, fortunately, these have not been seen to date. The first study I want to highlight is a phase 2A double-blind, placebo-controlled, randomized, multi-centering study designed to evaluate the safety, tolerability, and antiviral activity of Molnupiravir at a number of doses, dosed twice daily for 5 days in the treatment of individuals with mild to moderate COVID-19. Patients were eligible for participation in this trial if they were greater than or equal to 18 years of age and had tested positive for SARS-CoV-2 infection within 96 hours and had symptoms associated with COVID-19 for less than 7 days prior to the initiation of treatment. Antiviral activity, safety, and tolerability were assessed for 28 days following initiation of treatment. The presence of SARS-CoV-2 was assessed using nasopharyngeal swabs collected at baseline, days 3, 5, 7, 14, and 28. Safety was also assessed on these days, and adverse events were monitored throughout the study. Participants were randomized into the study 1-to-1 to Molnupiravir 200 milligrams or matching placebo, or 3 to 1 to Molnupiravir 400 milligrams or 800 milligrams or placebo. As mentioned, doses were administered orally twice daily for 5 days, and dose escalations did occur following review of safety and virological data from this and other studies. Regarding the endpoints, the primary endpoint was antiviral efficacy, measured as time to viral clearance, defined as the first time point when viral RNA was achieved that was below the limit of quantitation. A secondary antiviral efficacy endpoint or outcome was time to infectious virus elimination and median viral RNA change from baseline on days 3, 5, and 7. With respect to the demographics of the study populations, they were relatively comparable. The one thing that I will point out is in the groups between 70 and 90% of participants that were enrolled in the groups were white. Again, knowing that there are metabolisms of Molnupiravir to an active component, there may be concerns regarding some racial differences. So I just wanted to bring this to your attention. With respect to the results, the important thing to point out is that the greatest response rate and the shortest mean time to response were noted in the 800 milligram twice daily group. This group showed the greatest percentage of individuals with response and demonstrated, again, as mentioned, the shortest time to response. Looking at the Kaplan-Meier graph, again, this just highlights that around day 14, you can see the biggest deviation in the treatment groups and placebo, favoring the 800 milligram dosing regimen. Looking at genotypic changes in the viruses, the RNA-dependent RNA polymerases were evaluated, and was noted that there were changes that occurred at a higher rate among participants who received Molnupiravir compared to placebo. These data did support the hypothesis or the theory that viral aerial catastrophe was the primary mechanism of action. Regarding safety and tolerability, the incidence of treatment-associated adverse events was actually the lowest in the Molnupiravir 800 milligram group. The only adverse events reported by more than 4 participants were headache, insomnia, and increased alanine aminotransferases. So, in general, the medication was well tolerated. In conclusion, it was determined that Molnupiravir was well tolerated and produced significant reduction in infectious virus isolation, reduction in time to elimination of SARS-CoV-2 RNA, increase in the proportion of patients that cleared SARS-CoV-2 RNA, and reduction in SARS-CoV-2 viral RNA from baseline compared to placebo in outpatients with COVID-19. So again, not only did it appear that the patients benefit from clearance, but the patients also appeared to be less infectious when they received the Molnupiravir 800 milligrams. The MOVO trial was a phase 3 randomized placebo-controlled double-blind multi-site study of non-hospitalized adult patients with laboratory-confirmed mild to moderate COVID-19. Individuals had to have at least one risk factor associated with poor disease outcome and symptoms onset of less than 5 days prior to randomization. The primary objective of the MOVO trial was to evaluate the efficacy of Molnupiravir compared to placebo as assessed by the percentage of participants who were hospitalized or died from the time of randomization through day 29. Patients were eligible for participation if they were greater than or equal to 18 years of age, had laboratory-confirmed mild to moderate COVID-19, and symptom onset within 50 days of study randomization. Again, all patients were required to have at least one risk factor associated with poor disease outcome at study entry. On August 5th, there was a planned interim analysis that was conducted looking at data from 775 patients. At this time, owing to favorable results, study recruitment was halted. At this time, the trial was approaching full recruitment, for an estimated sample size of around 1,500 patients, and more than 90% of the intended sample size had already been enrolled prior to the determination to halt study enrollment. At the interim analysis, these were the data that actually generate a lot of excitement with Molnupiravir. At this point in time, there were 385 patients in the Molnupiravir group that were evaluated and compared to 377 placebo-treated patients. When we're looking at all-cause hospitalization or death through day 29 with the modified intent-to-treat analysis, there was a reduction in the risk of hospitalization or death by about 50%, and this was statistically significant. Additionally, there were no deaths in the Molnupiravir-treated group and 8 deaths in the placebo-treated group. So the risk of death was 100% lower in those treated with Molnupiravir. It was these data that caused the investigators to halt enrollment and proceed with emergency use authorization application. However, after a short period of time, the final analyses were released, and the data seemed to be a little less favorable. At this point in time, when we had 709 patients in the Molnupiravir group and about 700 patients in the placebo group, the relative risk reduction of all-cause hospitalization and or death through day 29 was only about 30%. So that was a little less dramatic than the 50% that had been seen on the interim analyses. However, of note, the risk of death was still appreciably lower in the Molnupiravir-treated group. There was 1 death in the Molnupiravir-treated patient population compared to 9 deaths in the placebo. With respect to safety and tolerability, the incidence of adverse events was comparable between Molnupiravir-treated patients and placebo, and the incidence of drug-related adverse events was comparable between the 2 groups. The most common adverse events noted were diarrhea, nausea, and dizziness. As I mentioned, the EUA for Molnupiravir was generated using the interim analyses, and when that was submitted to the FDA for review, about that time is when the final data were published. Based on the full data set, the FDA panel did not review incredibly favorably. However, an EUA was narrowly granted on November 30th by the FDA. Of note, in the United Kingdom, Molnupiravir had been approved for use or authorized for use on November 4th. With respect to eligibility for use, the criteria for use for Molnupiravir are for the treatment of mild to moderate COVID-19 in adults with a positive result of a direct SARS-CoV-2 viral test who are at high risk for progression to severe COVID-19, including hospitalization or death, and for whom alternative COVID-19 treatment options authorized by the FDA are not accessible or clinically appropriate. In terms of eligibility, again, these must be adult patients greater than or equal to 18 years of age, not pregnant or breastfeeding, have that positive test for SARS-CoV-2, and possess at least 1 risk factor for developing severe illness. And I'll cover those on the next slide. Of note, it's interesting that no criteria related to initiation of treatment within a window following onset of symptoms was included in patient eligibility. With respect to the risk factors for developing severe illness, you can see here, these are the risk factors that are listed or have been identified by the CDC. Those that have strong evidence, moderate evidence, and limited evidence. It's important to note that Molnupiravir is not authorized for use in patients who are less than 18 years of age. It's not authorized for the initiation of treatment in patients hospitalized due to COVID-19. And the reason for this is really it hasn't been studied in this way, and the benefit of treatment with Molnupiravir has not been determined. It's also not recommended that Molnupiravir be used for longer than 5 consecutive days. Again, maybe for the risk of some of the toxicities that had been seen in some of the animal studies. Additionally, it's not intended to be used as pre-exposure or post-exposure prophylaxis for the prevention of COVID-19. Just summarizing the adverse events that we noted for Molnupiravir, these are the common adverse events. Again, as mentioned, dizziness, headache, and diarrhea seem to be the most common side effects that are noted with Molnupiravir. However, it is generally well tolerated. Moving on to Paxlovid. Paxlovid is a combination of nirmatrelvir and ritonavir. Nirmatrelvir is a inhibitor of the SARS-CoV-2 main protease, Mpro, which is also referred to as a 3C-like protease. And an inhibition of this protease renders the enzyme incapable of processing the polyprotein precursors and thus preventing viral replication. It's important to note that currently Paxlovid is available in a card, and each card will contain 2 pills of nirmatrelvir and 1 pill of ritonavir, and these are to be taken together. With respect to pharmacokinetics, following a single administration of nirmatrelvir and ritonavir, AUC is about 23 micrograms hours per milliliter, and the Cmax is about 2.2 micrograms per milliliter. Each of the individual tablets of nirmatrelvir is 150 milligrams. When ritonavir and nirmatrelvir are co-administered, the primary route of elimination becomes renal through to the inhibition of enzymes, the CYP3A enzyme by ritonavir. So nirmatrelvir then becomes renally eliminated. As such, there is emerging evidence that suggests that dose reductions might be necessary for individuals with severe renal impairment. Nirmatrelvir, co-packaged with ritonavir, is an inhibitor of cytochrome P453A, and thus may increase plasma concentrations of drugs that are metabolized by cytochrome, the cytochrome isoenzyme system. Additionally, induction of CYP3A may result in decreased plasma concentrations of nirmatrelvir and ritonavir. Additionally, nirmatrelvir is a substrate inhibitor of P-glycoprotein. So taking all this together, it's evident that there is a significant risk or likelihood for drug-drug interactions with Paxlovid. The half-life of the nirmatrelvir is approximately 6 hours. In a phase 2/3 evaluation of protease inhibition for COVID-19 high-risk patients, or the EPIC-HR trial, patients were randomized and were evaluated in a double-blind manner. Individuals were non-hospitalized adult patients with COVID-19 who are at risk of progression to severe illness. This trial was initiated in July of 2021. So at that point in time, there was likely to be alpha and delta type variants. The primary analysis of the interim data was set to be evaluated following the enrollment of roughly 1,200 adults, and these were enrolled by September 29th. The medications were given together twice daily for 5 days. In the final analysis, there were roughly 2,200 patients that would have been enrolled, and it was revealed that there was an 89% reduction in the risk of COVID-19 related hospitalization or death compared to placebo for individuals who were treated within 3 days of symptom onset, which was their primary endpoint, and 88% of patients treated within 5 days of symptom onset, which was their secondary endpoint. So essentially with this, they found no benefit from treating early with the Paxlovid compared to the 5 days of onset. important to note that less than 1% of patients who received Paxlovid were hospitalized through day 28 following randomization, compared to 6.3% of the patients who received placebo. There was a relative risk reduction of negative 5.6, and this was statistically significant. The relative risk reduction increased to 94% when we looked at patients who were greater than or equal to 65 years of age. In the overall study population through day 28, no deaths were reported in patients who received Paxlovid, compared to 12 deaths among those who received placebo. The SARS-CoV-2 viral load was assessed at baseline and at day 5, and those data have been evaluated for about 500 patients. In this analysis, Paxlovid was determined to reduce viral load by approximately 10-fold compared to placebo. Treatment emergent adverse events were comparable between placebo and Paxlovid, and few serious adverse events and study discontinuation were observed in either of the arms of the study. A second trial, EPIC trial, was also conducted, and the data were available. This was for standard risk patients. So these were individuals, including unvaccinated adults, who are at standard risk or low risk of hospitalization and death, as well as vaccinated patients who had one or more risk factors for progressing to severe illness. We saw similar endpoints here for this trial. An interim analysis of the standard risk patients was performed when 80% of patients had been enrolled in the study. At this time, no difference in the primary endpoint, which was the self-reported sustained alleviation of all symptoms for 4 consecutive days. And for the secondary endpoint, there was less than 1% of those who received Paxlovid were hospitalized following randomization, compared to 2.4% of individuals who had received placebo. This difference did not reach statistical significance. However, there was an approximately a 10-fold reduction in decrease in viral load compared to placebo. And this finding of a reduction of viral load was consistent from the EPIC-HR study. Based on the data from the EPIC trials, Paxlovid was reviewed and granted its EUA on December 22nd of 2021. The intended use for Paxlovid is for the treatment of mild to moderate COVID-19 in adults and pediatric patients 12 years of age or older weighing at least 40 kilograms, as long as they had a positive result with a direct SARS-CoV-2 viral test and who are at high risk for progression to severe COVID-19, including hospitalization or death. Patient eligibility, again, adults or pediatrics greater than 12 years of age and weighing at least 40 kilograms. Positive test for SARS-CoV-2. If treatment was initiated within 5 days of symptom onset, so here, unlike we saw for Molnupiravir, we are being recommended to follow a treatment window, and these individuals possess at least one risk factor for developing severe illness. Of note, the Institute for Safe Medication Practices, subsequent to the EUA authorization, did put out a warning regarding renal impairment. For those with moderate renal impairment, it's recommended that pharmacists remove one of the Molnupiravir tablets from both the morning and evening doses from the blister card before it's dispensed, and cover the hole with a manufacturer-supplied sticker. It's also recommended that individuals with severe renal impairment not receive Paxlovid. Additionally, Paxlovid is not authorized for initiation of treatment in hospitalized patients due to severe or critical COVID-19. It's not authorized for pre-exposure or post-exposure prophylaxis, nor is it recommended or approved, or authorized rather, for use for longer than 5 consecutive days. In summary, Molnupiravir and Paxlovid both now have received emergency use authorization for the treatment of SARS-CoV-2 infections in outpatients who have mild to moderate COVID-19 at high risk for progression to severe disease. Paxlovid does appear to be a slightly more promising agent, but it does have high risk for drug-drug interactions. EUAs were granted for the treatment of infective outpatients again, and those were high risk for progression to severe illness. For those with standard risk, it does not appear that there is a clinical benefit for those individuals. Unfortunately, owing to language in the EUA, pharmacists are not able to independently prescribe either of these agents in spite of being granted authorization under the PrEP Act. Therefore, if pharmacies or pharmacists are going to engage in test and treat models, they will be required to obtain a CPA or authorization via another mechanism for these pharmacy test and treat models. I hope you found the information that was presented today helpful, and stay safe. Thank you.